that okay? All right. Let's go then. Before we start on the metabolism of lipids, are there any questions that people have about photosynthesis, the topic we finished up on Friday? Any questions about photosynthesis? Okay. Well, then let's get going. We've done one major big area of metabolism by talking about carbohydrates, gluconeogenesis, glycolysis, glycogen synthesis and breakdown. We talked about the kind of meeting point of many different kinds of metabolism when we talked about the citric acid cycle. We first met it through the carbohydrates, but we sort of mentioned the fact that we were going to meet it again and again. And now we're about to begin the second big area of metabolism. And in some respects, you may find it simpler than carbohydrate metabolism. There's not as much stuff happening along the backbone of many lipids. And in fact, there's some biochemistry textbooks that start with lipid metabolism rather than with carbohydrates. But it's an area of biochemistry that should be of very active interest to all of us. Current estimates are that roughly half the people dying in the U.S. die from atherosclerosis, which is essentially a disease of lipids. It's the hardening of the arteries where you deposit cholesterol and LDLs in the lining of your arteries to the point where you can't get blood through to supply the heart or the brain. And that's sort of a form of rampant, out-of-control lipid metabolism. But by understanding ordinary lipid metabolism, you'll have a better context for understanding atherosclerosis. Another reason to be thinking about lipid metabolism is that many serious public health people think of obesity today as an epidemic, that the levels of obesity in the United States have risen. They appear to be continuing to rise, and quite apart from any difficulties that obesity presents just from a practical day-to-day point of view, it gives rise to a number of other diseases as well. So things like diabetes and arthritis and things like that are very seriously impacted by having obesity. This is sort of an overview for you of some of the higher level aspects of lipid metabolism and how it relates to these things. So in the intestine, we process food in the stomach and part of the intestine, then we have to absorb it. Lipids are absorbed in the form of something called chylomicrons, which are in the bloodstream and can actually make the blood look milky if you have enough fat in a meal. The chylomicrons are transported by the blood to a couple of places, to the liver certainly, which is going to package and export the lipids in them in the form of something called VLDL, stands for very low density lipoprotein. And they're very low density because they have a lot of lipid in them. Some tissues can take chylomicrons directly. But the VLDLs can be exported into the system, interacting with tissues, depositing some of their lipids in those tissues, and therefore becoming not so light. They become just simply low-density lipoproteins, which can also be used. And these get circulated to the liver. If enough lipid is taken out of them, they become HDL, which are high-density lipoproteins, and they are further metabolized by the liver as well. To give you a feeling for how serious atherosclerosis can be, this is a cross-section through an artery stained for light microscopy, and you can see that this is all the hollow space that's left. The original artery lumen or space was this big, and all of this colored stuff is blocking the way. So you can imagine it's hard to get as much blood through that as it would be through the native, clean, healthy vessel. So thinking in metabolic terms, this is sort of our high-level roadmap again. Our polymer in this case is not a very big polymer. It's the triacylglycerols. Those are broken down into monomers of fatty acids. We're going to see that fatty acids are broken down further into 2 and 3 carbon fragments. That degradation is principally for energy. Those 2 and 3 carbon fragments are fed into the citric acid cycle. We can synthesize fatty acids. We can also synthesize a variety of other lipids, and we'll talk about some of those as well. Another metabolite of the fatty acids is ketone bodies. Ketone bodies are not something that we generally have in abundant levels all the time, but they're synthesized when we're fasting, when we're not taking in food on a regular basis and maintaining the blood glucose level that way. And ketone bodies are used by tissues during fasting, especially by the brain. And I'll have some things to say about that in a little bit. There are some other lipids besides fatty acids, of course. We talked about some of those earlier when we talked about the structure of lipids and membranes, and we'll speak to that a bit. We're going to speak a little bit about the fact, which is actually a little weird, that lipids can act as biological signals, that some subset of the lipids in our bodies are actually modified in ways that let them be signaling molecules. And then talk a little bit about cholesterol metabolism, very different from fatty acids, but also very important in its own right. So, give you a little feeling for where we're going. Now, fatty acids degradation is a wonderful source of energy. Fatty acids are mostly reduced, so you get more energy out of fatty acids than out of carbohydrates. Roughly speaking, fat has twice the energy density of carbohydrates. That's why migrating birds use it. A migrating hummingbird that tried to use glycogen to migrate would be too heavy to get off the ground. But as triacylglycerides, you can't really use them. That's a storage form. And so you have to get the fatty acid out of the triacylglycerides. And when you're talking about the dietary triacylglycerides, getting them in from your food source, there are lipoprotein lipases on the cell surface that allow for the selective nicking off of fatty acids from the triacylglycerides in order to bring them into the tissue to feed that tissue. There are other lipases, again, lipase, something that splits lipids, that are hormone sensitive. And these are lipases that are in the tissues that store triacylglycerides, such as adipose tissue. And that hormone control allows the body to then release the fatty acids into the circulation when you need to feed other tissues. Fatty acid by itself is not found free to any great extent. In the bloodstream, for us at least, it is generally carried by albumin. Albumin is this sort of nondescript looking blood protein that turns out to bind half the things that we think about. But it's certainly one of the things it carries is free fatty acids. If we didn't keep the levels of free fatty acids down, it can cause structural damage. Free fatty acids are actually a detergent, if you think about them. They've got a charged head and a nonpolar tail. And this is one reason why olive oil is actually a decent antibacterial agent. There's several % free fatty acids in most olive oils. If you've ever wondered about the Good Samaritan who poured oil in the wound, it's because olive oil is an antibacterial. When you don't have triclosan and things like that, you use what you have to hand. And this is just showing you that in structural terms. You have your 3-carbon glycerol backbone. This is a triacylglycerol, each with an acyl tail there. And it's a free reaction. It's just a hydrolytic one. So you get back a glycerol molecule. And that glycerol molecule can be used for quite a variety of things. We'll talk about that at various times. And here's your fatty acyl group that you can use for energy. Okay. But you can't just simply take up that fatty acid and use it. You've got to somehow or other activate it in a way that allows you to transport it to the part of the cell where you're actually going to metabolize it. And of course, that does take a little bit of energy. So there's an energy investment phase there. You have your fatty acid. ATP is going to make it into an acyl adenylate. So this is the acyl group here. It was a triacylglyceride before. Now it's an acyl adenylate because you have adenosine monophosphate in there. And because the inorganic pyrophosphate is split to 2 pyrophosphates, that helps drive the reaction in this direction. But this is now a high energy bond. And that high energy bond allows you to transfer the acyl tail to a CoA molecule. So now you have an acyl CoA and an AMP molecule. So it costs 2 high energy bonds to activate a fatty acid into a form that can be then carried around and used within the cell. That R there can be many different things. It might be 16 carbons, it might be 18 carbons. The machinery doesn't really notice that. But the place where we're going to actually want to get that acyl adenylate, or the activated fatty acid, is into the matrix of the mitochondrion. The degradation of fatty acids is going to occur inside the mitochondrion in the matrix where all the wonderful reduced cofactors that it produces is right there for electron transport. It's a very efficient place to do it. But you can't get that acyl adenylate into the mitochondrion in that form. And so we have a carnitine transporter in the mitochondrial membrane. The acyl-CoA transfers the acyl group to carnitine. And if you look at this molecule, you can see it's kind of a funky amino acid. It's got an amino group here. Admittedly, it's a tertiary amine, and it's got an acid there. So some people refer to carnitine as an amino acid. Strictly speaking, they're correct. It's not one of the amino acids we use to make protein, of course. But it can form an acyl carnitine derivative like this that has an equivalent energy to the acyl-CoA. So the carnitine transporter recognizes the carnitine moiety, and it will pull through the acyl part independent of whatever that acyl group is. And once it's in the mitochondrial matrix, the reverse reaction happens. You have a CoA come in, receive the acyl group, so you've regenerated your acyl-CoA, and now the carnitine is carried back out into the cytoplasm where it can pick up another molecule of an acyl group. This carnitine transporter is pretty important, and so is carnitine. Carnitine, you might guess, gets its name because it was isolated from meat. That carn root comes from meat. And if people have deficiencies in carnitine, they have a hard time getting fatty acids into their mitochondria. The symptom of this is something called claudication or intermittent claudication, which means your legs hurt when you walk, not your feet, but your calves, the back part of your muscles. Because if they can't get the fatty acids into the mitochondrial matrix, they start running short of energy. So now we've got our acyl-CoA there in the matrix of the mitochondrion, and what are we going to do? We're going to break down most of the fatty acyl groups through beta oxidation. This is the principal route. When fatty acyl groups are broken down this way, it's principally for energy production. We're going to feed the products of it into the electron transport chain. And it happens, oddly enough, at the beta carbon. So beta oxidation happens at the beta carbon. If this is the carbonyl carbon, the first one is the alpha carbon, the second one is the beta carbon. So what we're going to see is multiple rounds of oxidizing that beta carbon. And each round of beta oxidation produces a molecule of acetyl-CoA. So here's a 2-carbon fragment, here's another 2-carbon fragment, here's another 2-carbon fragment, and so on. The beta oxidation is going to happen at the activated end, the end nearest the CoA compound. In principle, you could have started at this end. Chemically speaking, very difficult. So the beta oxidation is working its way in from the CoA end of the molecule. And we're going to see that beta oxidation has 4 reactions that are actually quite similar to reactions you've seen before in the context of the citric acid cycle. So here we have fatty acyl-CoA. Here's the carbonyl carbon, the alpha, the beta carbon. Okay? This is the carbon that's going to get oxidized. And the first reaction is acyl-CoA dehydrogenase, producing an FADH2 and a QH2. So the electrons from that carbon are being transferred first to FAD and then to Q. So you're going to produce some of the lipophilic electron carrier QH2. And then the result of that is, instead of a plain old single bond there, you now have a double bond there. This is an enoyl-CoA because of the double bond. Hydratase can add water across that. So this is what we do, to refresh your memories, what we're talking about are reactions that resemble the reactions from succinate, fumarate, malate, oxaloacetate. So we're going to be adding water now to that double bond. Now we have 3 hydroxyacyl-CoA, so it's 1, 2, 3 carbon, the 3rd carbon in. So 3 hydroxyacyl-CoA. Should look a little familiar. And here we have hydroxyacyl-CoA dehydrogenase using NAD+ as the electron receiver. So we're going to oxidize this yet further. We've stripped off some more electrons. We now have a keto-acyl-CoA, which should remind you kind of oxaloacetate. And then the next step is a special enzyme called thiolase that can bring CoA in and this arrangement of two carbonyl carbons that are kind of near but not next to each other are capable of releasing this molecule and transferring that carbon onto the CoA molecule. So this carbon that we've just finished oxidizing to a carbonyl carbon is the first carbon now in the new molecule, the new fatty acyl-CoA. This is 2 carbons shorter than it was, and here is a molecule of acetyl-CoA that's been released, and that acetyl-CoA can go into the citric acid cycle, get burnt, and so on. So beta oxidation produces some QH2 and NADH in the oxidizing of that carbon, and then it produces an acetyl-CoA, which of course produces NADH and FADH2 as it oxidizes the acetyl-CoA. Any questions? It's an extremely productive way of getting energy. So for every 2 carbons in a fatty acid, you'll have 1 round of beta oxidation, producing a QH2, an NADH, and an acetyl-CoA. In the citric acid cycle, those guys are going to produce 3 NADH, a QH2, and a GTP. So when you start looking at all of the all the products of phosphorylation, oxidative phosphorylation, you get 3 ATP off of that first QH2, 2 ATP there, 3 from the NADH, and then the acetyl-CoA is going to provide 12 there. So you get 17 ATP for every 2 carbons in a fatty acid. And if you remember, we said that an entire glucose molecule gave you in the order of 32 to 34. That's 6 carbons. So you can see that this is doing roughly 50% better than that. Some fatty acids are a little bit more complicated. Some of them, for instance, have double bonds in them. And unsaturated fatty acids like this, if they have cis double bonds, they have to be rearranged into trans double bonds because the beta oxidation machinery works on trans double bonds, not on cis double bonds. One way of taking care of that is to reduce the double bond. So you're investing in NADPH here. You've reduced it to a single bond, and then the isomerase is free to rearrange things. In this case, it's rearranged the double bond to lie from between the 3 to 4 carbon over one bond worth to the 3 to 2 carbon. So now it has a double bond in a place where it would normally occur in the process of beta oxidation. And here's an isomerase working that had a cis double bond in this acyl group, and by the isomerase, we've rearranged it into a trans double bond that's 1 bond over. And now that would be in the right place to go ahead and do what we want to do during beta oxidation. Most of the fatty acids that we've talked about so far are fatty acids that come in multiples of 2 carbons, 16, 18, 22, 24. And when we talk about fatty acid synthesis, you'll see why it's so common to have multiples of 2. But there are some plants and some bacteria that make fatty acids that have an odd number of carbons. And so if you have an odd number of carbons, the beta oxidation machinery will work on it, chipping off 2 at a time until it gets down to a 3-carbon piece. And so here is what that 3-carbon piece looks like. It's propionyl-CoA, methyl ethyl propyl, propionyl-CoA. So that's not something that goes well into the rest of the machinery. So the cell invests a little bit of energy and adds a carbon to it at the cost of an ATP. So this is propionyl-CoA carboxylase, adding a carbon. Now we have a 4-carbon fragment. It's a branched one, but that's not a problem because by using a racemase, we can rearrange that around and generate succinyl-CoA. Now, the process of moving around a carbon is, chemically speaking, a little bit interesting. It's a little bit challenging. And one of the best cofactors for doing that is something called cobalamin cofactor. mutase uses that for rearranging the carbon atoms. This is what cobalamin cofactor looks like. You can probably recognize an adenine up there and another adenine down here. You can probably recognize a heme ring in here, and here's a cobalt in there, and spinach of various kinds around the edges. Cobalamin takes on the order of 12 reactions to be synthesized. It's derived from B12. We cannot make cobalamin by ourselves. We take it from B12. And it's one of the very few places where there's a carbon-cobalt bond. It's a very unusual chemistry to go from carbon to cobalt. Now, we've talked so far about beta oxidation, and beta oxidation takes place in the matrix of the mitochondria, and it's a principal source of energy for fuel. But there are some other places where fatty acids can be oxidized, and that's in a structure called a peroxisome. This is a trend, I think this is an electron micrograph that's been colored, picture of a peroxisome. You notice that it has a single membrane around it, in contrast to the mitochondrion, and it's got a fair bit of electron density to it, so it's got a lot of stuff in it. And some fatty acids are oxidized actually in peroxisomes, in contrast to the mitochondria. And the outcome of that is rather different than what we see in mitochondria. You start with an acyl-CoA again, but the acyl-CoA oxidase uses FAD as the receiver, and then is coupled to other reactions that will produce hydrogen peroxide. Hydrogen peroxide. Hydrogen peroxide is not exactly a friendly molecule. In some cells, such as certain kinds of blood cells that are involved in the immune defense, they can locally generate hydrogen peroxide at the site of an infection or something and use that hydrogen peroxide to degrade, to attack an organism. It's also a very nonspecific breakdown agent, so cells can use it to attack molecules that they don't have degradation machinery for. So if they've eaten up something that they don't know how to cope with it, a few rounds of hydrogen peroxide will usually reduce it to something that they can manage. After acyl-CoA oxidase, you then have an enoyl-CoA, and as you can see, that's something that can be further broken down. One of the important aspects of the peroxisome is that its machinery can handle branched lipids. So here is phytonate, which is a branched fatty acid. If you may remember that sort of spinachy stuff hanging off the sides of the chlorophyll molecule, that's phytonate, and mitochondria don't break that down, so you need to have peroxisomes to do that. People who are missing peroxisomes suffer from a variety of diseases if they can't do this. So although it's not the major route, it's nonetheless an essential route. Okay. So far, we've talked about breaking down fatty acids. We learned in carbohydrate metabolism that breakdown and synthesis sometimes have resemblances, can even sometimes share reactions. So when we talk about fatty acid synthesis and compare it to fatty acid breakdown, what you're going to have your eyes open for here is looking for similarities at the level of the carbon backbones, but there'll be differences in almost everything else. So this is not like glycolysis and gluconeogenesis, where they can share 80% of the pathway. These are really separate pathways in different parts of the cell. They have different enzymes. They have different cofactors, different costs. And an important component of the fatty acid synthesis is going to be something called acyl carrier protein. And I'd like you to take a little moment to look at the structure of that. We talked several weeks ago now about the structure of coenzyme A, that it has a business end of this thiol there. That's why it's always either SH or S, whatever's added onto it. But there's stuff in between. This is the structure of coenzyme A altogether. And this part of the molecule here is pantothenic acid, and it's a derivative of vitamin B5. Well, what's interesting about fatty acid synthesis is that the acyl carrier protein also has that complicated functional group covalently attached to the protein. It's like it has a covalently attached CoA molecule minus the ADP. The ADP was just acting as a zip code or a tag anyway, so covalently attaching it to the protein allows acyl carrier protein to have the chemical functionality of CoA as a permanently attached part of it. So we talked about beta oxidation, the breakdown of fatty acids in the mitochondrion. Synthesis of fatty acids is going to turn out to happen in the cytoplasm. And so somehow or other, we've got to get carbon fragments out to the cytoplasm for synthesis to happen out there. It turns out that most of the excess acetyl-CoA, so CoA you don't need this moment, this very second, for energy production, most of the excess acetyl-CoA is going to come from sugars or from amino acids in the mitochondrion. Obviously, if you have excess fat in your diet, you don't need to synthesize fatty acids out of that. You just deposit that. So when you're synthesizing fatty acids, you're synthesizing them from excess sugar, excess amino acids. And so you need some way to get the CoA molecules, acetyl-CoA molecules, out to the cytoplasm where the synthesis is going to happen. And frequently that excess is in the form of citrate. The citrate can be transferred through a transporter from the matrix to the cytoplasm. And here you have your citrate molecule. Those two carbons are the ones we're going to target. We spend a little bit of energy, here's an ATP invested, to make acetyl-CoA that releases the oxaloacetate, which will end up getting back into the mitochondrion. Now, a key enzyme in fatty acid synthesis is an enzyme called acetyl-CoA carboxylase. From its name, carboxylase, you can guess we're going to add a CO2 to the acetyl-CoA. This is the rate-limiting step for fatty acid synthesis. And in order to have this happen, you have to activate a biotin molecule with a carbon dioxide, and that's going to cost energy. So in order to add the bicarbonate to the biotin, we invest ATP, so it's one high energy bond to do that. And this biotin has a sufficiently energetic bond that we can transfer that now onto the acetyl-CoA, and now we have malonyl-CoA. Malonyl-CoA is the donor of the two carbon fragments that we're going to add on during fatty acid synthesis. In beta oxidation, we produce two carbon fragments in the form of acetyl-CoA. During fatty acid synthesis, to add two carbons at a time, we need an activated form of it, so to speak. And malonyl-CoA with this disposable carbon dioxide is the form that's going to turn out to be useful for that. Fatty acid synthase is kind of a nifty protein. This is an electron micrograph of fatty acid synthase. And in some species, a single polypeptide has all 7 enzymatic activities that are needed in order to synthesize fatty acids. The first step that we're going to need to do is to prime the enzyme and get something on there to get the process started. We have in the resting state, if you like, there's a cysteine that has an acetylcysteine on it. Acyl carrier protein can transfer the can attack rather that 2 carbon fragment so that these 2 carbons are going to get added to this carbon, and the terminal carboxylic acid is going to be lost as carbon dioxide, and so that's going to then generate now a 4-carbon acetoacetyl ACP. So the loss of carbon dioxide here is what powers the addition of those 2 carbon fragments. So we've got incoming acetyl-CoA, and that's used to charge that cysteine so it has a 2 carbon fragment on it. We've got an incoming malonyl-CoA, the 3 carbon fragment, and it's going to be transferred by a transacetylase to the acyl carrier protein. So transacetylase can catalyze both of these reactions because they're energetically equivalent. One charges the cysteine with the 2 carbon fragment. The other charges the acyl carrier protein with the malonyl fragment. And then the condensation between those two groups is ketoacyl synthase. And now you have acetoacetyl acyl carrier protein. So this is one acetyl group and a second acetyl group, so it's an acetoacetyl acyl carrier protein. And this should look a little bit familiar. This is a very oxidized carbon. If we're making fatty acids, an oxidized carbon is not what we want. What we want is a reduced carbon. So we can guess that some of the energy and attention that's going to go into this molecule is going to be focusing on reducing that carbon down to a lower reduction state. So here we have an NADPH. The NADPH is the most common reduced factor that's used for biosynthesis. The P is just simply a marker, doesn't change the energy of it at all. It just says, use me for biosynthesis. And now we've reduced that carbonyl carbon to an alcohol. This is hydroxybutyryl acyl carrier protein. One thing I want to point out to you is we've got intermediates here that look rather similar, but they're not identical. So when we're doing fatty acid synthesis, we have this 3-hydroxy acyl carrier protein with the alcohol up. And when we were doing beta oxidation, we had a 3-hydroxy intermediate that had the alcohol down. And so the overall structures are the same in synthesis and degradation, but the configuration, whether it's a D or an L configuration, is different between the two processes. So we're not all the way there yet. It's partially reduced. We want to reduce it some more. There's a dehydrase that takes the water out and generates the double bond. And so now we have a butenoyl acyl carrier protein. It's not quite fully reduced yet, and so you invest another NADPH and reduce it down to butyryl acyl carrier protein. So now we have a 4-carbon acyl group with the carbonyl carbon adjacent to the acyl carrier protein. This group looks a lot like the acetyl group that was on the enzyme cysteine group. This one can be transferred by transacylase to that group, and then the acyl carrier protein can receive another malonyl CoA in to charge the acyl carrier protein with a malonyl group. So whereas the beta oxidation started at the carbonyl end and cut off 2 carbons at a time, the fatty acid synthesis is going to add on 2 carbons at a time, but driving that addition through the loss of the carbon dioxide from the malonyl group. And so as you repeat this 8 times, you're going to build up a palmitoyl acyl carrier protein. And the fatty acid synthase, to some extent, can recognize the size of the molecule. So the palmitate is the principal size that it's targeted for. It's not perfectly good at counting. And is there a problem? Sorry, okay. This can be recognized as the correct length. A thioesterase can simply split that bond with water. And so we're going to throw away the energy implicit there. And now we have a palmitate molecule, a free palmitate. So, any questions? Well, palmitate is not the only thing that we need. Let me just back up one. So we have, right. Palmitate is not the only molecule that we need. We sometimes need longer molecules. We sometimes need desaturated molecules. And there are enzymes that can do all of those things. So elongases, no surprise, add 2 carbon fragments. Desaturases are capable of creating a double bond. These enzymes are in a couple of different places. The endoplasmic reticulum has some of them. The mitochondrion has some of them. We mammals are limited in what we can do. We cannot go past the C9 to make a linoleate. So here we have the molecule of linoleate, which has 2 double bonds. And here we have linolenate, which has 3 double bonds. If you start counting the carbons, you have 1, 2, 3, 4, 5, 6, 7, 8, 9. Here's the 9 carbon. We can do this we can't do this. And since we can't make that second double bond, we're dependent on linoleate and linolenate in the diet. If we get them in the diet, we can desaturate to linolenate. We can, to some extent, we can elongate. If you look here, you'll notice that here are these 3 double bonds sort of near each other. It's longer here now than it was there. So the elongation happened at that end of the molecule. And then this longer molecule can be further desaturated until you have 4 double bonds. This is arachidonate now. And we're going to see that arachidonate can do a lot of rather cool things. So there are a number of ways of naming lipids. One is counting in from this end. So this would be C9, counting in from this end, starting at the carboxylic acid. You can also count in from the end, or omega, 1, 2, 3, 4, 5, 6. And so in that nomenclature, this would be called an omega-6 fatty acid. You may have heard of omega-6 fatty acids as one of the essential fatty acids. There are also omega-3 fatty acids in which 1, 2, 3 carbon is the carbon for a double bond. And those are also essential. We cannot make them either. Interestingly, speaking about these double bonds and stuff, the way in which they found out about the essential character of essential fatty acids was that they discovered during the 19th century, in the Victorian heyday of exploration, that Arctic explorers who lived on Arctic hares, the sort of rabbit-like animals that live in the Arctic, could die of a nutritional deficiency. They didn't look sick in general, they just up and died. It turns out that Arctic hares have so little body fat that they did not have enough essential fatty acids to support life if they were the only thing you were eating. It was called rabbit starvation. So you needed to eat something else as well as rabbits if you wanted to survive in the Arctic. Well, how do we control all this? This is obviously a pretty energy-dependent process. We're investing ATP at several steps. We're investing NADPH. We got that NADPH from the pentose phosphate pathway. So it's worth seeing how we control fatty acid synthesis. Fatty acid synthesis and fatty acid breakdown are obviously opposite processes. You would really like to be able to make sure that you're not doing very much of one if you're doing the other. And so the regulation of these processes is going to reflect that fairly well. Let me just see something else. Okay. These pathways have regulation at a number of different levels. As you can imagine, anything that is this energy dense is going to involve attention to it at both the very local level and the whole organism level. So some of the places where we see controls are committed steps, as we saw in glycolysis, for instance. There's regulation of transport, which in a sense, carbohydrate metabolism is regulated because glucose uptake is regulated. There's local controls. There are also global controls. So if I'll remind you that there are 2 kinds of transport processes that we looked at. There was a carnitine transporter that was a way of taking fatty acids into the mitochondrion as a fuel for beta oxidation. We also looked at the citrate transporter as a way of transporting citrate out as a way of providing acetyl-CoA to the cytoplasm for fatty acid synthesis. So those are the 2 transport steps that are going to be of interest. We talked about the synthesis of malonyl-CoA as a committed step, that acetyl-CoA itself can be used for several things. And so making malonyl-CoA, the malonyl-CoA is only going to get used by the fatty acid synthase. So that's a committed step that's worth regulating. And so let's look at how these different metabolites interact with the 2 different overall processes to turn them in the right direction at the right time. So citrate stimulates acetyl-CoA carboxylase. So citrate, which is a precursor of that acetyl-CoA, is a positive regulator to make more malonyl-CoA. So high levels of citrate are saying to the cell, energy is abundant, save some of this for a rainy day. Presumably you're going to go on making fatty acids and so forth. At some point, your levels of fatty acids are rising. you've made enough, or maybe you've eaten a meal that has a lot of fatty acids. Fatty acids, by contrast, inhibit the committed step. Again, a fairly logical kind of feedback inhibition. It's kind of a downstream feedback, right? Because a complete fatty acid is the end point of many, many chemical reactions. So fatty acids will inhibit acetyl-CoA carboxylase. But we also see regulation at the level of transport. So malonyl-CoA, which is the product of the committed step, it's only going to be useful for fatty acid synthesis. It ensures that we're not going to waste the fatty acids by sending them into the mitochondrion to burn them up. It inhibits the carnitine transporter so that the fatty acids that are made by fatty acid synthase are not shuffled off into the mitochondrion to be burnt up and waste all that effort. So these are, you could consider this the most local of controls where you have metabolites feeding back. You're able to integrate the different compartments of the cell because the malonyl-CoA is ensuring that the fatty acids aren't going to be sent where the other process is active. But you also have regulation at the level of the whole body through hormonal control, and we'll probably talk about some of those later on as well. Okay. And maybe this is a good place to take a break. Let's see. I think this is probably a good place to take a break. AV, can we cut?